Standard operating procedure for micropipettes. There are different types of micropipette such as the 10, 20, 100, 1000 and 5000 microliter micropipettes. Here we will see the different types of tips for each micropipette. Generally, there are 3 types of tips. The white tips are for the 10 microliter micropipettes. The yellow tips are for the 20, 100 or 200 microliter micropipettes. The blue tips are for the 1000 microliter micropipettes. The big white tips are for the 5000 microliter micropipettes. First, we will look at the forward pipetting technique. This technique is for aspirating standard solutions such as water, buffer or diluted saline. Turn the adjusting knob to the desired volume. Do not exceed the maximum or minimum volume stated on the micropipette. Insert pipette tip onto tip cone. Generally, there are 2 plunger stops for all micropipettes. Push the micropipette plunger until the first stop. Immerse pipette tip into the liquid, then release the plunger slowly until the tip fills up. Then dispense the liquid by pressing down the plunger to the first stop and the remaining liquid by pressing the plunger to the second stop. Remove the tip into the waste container by pushing the tip ejector. Reverse pipetting is done by pushing the plunger to the second stop. Immerse tip into the liquid. Release the plunger slowly until the tip fills up. Dispense liquid by pressing down the plunger to the first stop. Dispense the remaining liquid to the original container or discard. This technique is used commonly for aspirating viscous solutions. Finally, always remember to readjust the volume of the micropipette to the maximum when your task is completed. Common mistake made by micropipette users include pushing down the plunger to the second stop for non-viscous solutions. Here you can clearly see the difference in the volume due to the pipetting error. Most importantly, do not place micropipette with fill tip horizontally on the bench to avoid cross-contamination.